Welcome to GRS Learn Life Sciences YouTube channel, Cell Cycle Regulation Part 2 in English. This video is highly useful for who are writing CSIR, GATE, ICMR, DBT, AP and other states, SET, IIT JAM, IIC, PG and PhD entrance test, all central university entrance tests, APRESET, APPSC DLs, APPSC JLs and NEET exam. If you like this video, please subscribe, share, like and comment.
This is second part of eukaryotic cell cycle regulation. If you have not watched my first video, please watch that video to understand eukaryotic cell regulation completely. Hi viewers, in this video first I am going to explain discovery of MPF, maturation promoting factor, how researchers identified or discovered this maturation promoting factor. Next I discuss discovery of cyclins. Cyclins are proteins which are important for MPF activity. Next I discuss structure of MPF. MPF complex is made of cyclins and CDKs. I am going to explain that MPF structure. Next I explain CDKs and cyclins in cell cycle regulation, how MPF regulates different phases of cell cycle, that I am going to explain. Then I explain regulation of CDKs by inhibitors, inhibitory phosphorylation, cyclins and proteolysis. These factors they regulate CDKs and thereby regulating cell cycle. Finally I discuss regulation of CDKs and cyclins by external signals and internal signals. In my first video I explained what is external signal and what is internal signal, and how these external signals and internal signals they regulate cell cycle by regulatory point and checkpoints in G1 phase, S phase, G2 phase and M phase of cell cycle. Let us start with discovery of MPF. This MPF is identified by two scientists, the Saccharomyces and Clement Market. These two researchers, this MPF works at regulatory point of cell cycle. In eukaryotic animal cell and yeast cells, the regulatory point is present between G1 to S, and this regulatory point allows cell to enter into S phase from G1 phase. In some cells such as fission yeast and oocytes, the regulatory point is not present between G1 and S phase, it is present between G2 and M phase. In oocytes, the external signal that cell to enter into M phase from G2 phase is progesterone. In case of animal cells, the external stimulus is growth factor. In case of yeast cells, external stimulus has three factors: cell size, nutrients and mating factors. Cell size, mating factor and nutrients makes cell to cross restriction point. In case of oocytes, to cross that restriction point, oocytes need progesterone hormone. So in presence of progesterone hormone, cell enters into M phase from G2 phase. And these researchers they took cytoplasm of M phase cell of oocyte and injected this cytoplasm into another oocyte which is in G2 phase. After injection of this cytoplasm into G2 phase of another oocyte, that oocyte progressed into M phase by crossing the restriction point. Researchers found that cytoplasm has some factors, that factors making cell to enter into M phase from G2 phase in oocytes, and that factor is called maturation promoting factor. And this is how researchers identified this maturation promoting factor, which makes cell to enter into M phase from G2 phase in oocytes. In yeast cells there is another regulatory protein, that protein is called CDC protein, and let us see how this CDC protein is discovered in yeast cells. Researchers made CDC temperature mutants, permissive temperature and non-permissive temperature. In permissive temperature, these cells they can synthesize CDC protein. At non-permissive temperatures, these cells they cannot synthesize this CDC protein. Since at non-permissive temperatures the cell cannot synthesize CDC protein, this yeast cell cannot be divided. But cells at permissive temperatures they can synthesize this CDC protein, therefore the cell can complete cell cycle and will produce daughter cells. Researchers identified that this CDC protein is very important in yeast cells to cross the regulatory point and to enter into S phase from G1 phase. Genetic analysis of MPF. So after identifying maturation promoting factor in oocytes and CDC protein in yeast cells, researchers analyzed its genetical nature. In Saccharomyces cerevisiae, which is a budding yeast, the regulatory protein is CDC28. In Saccharomyces pombe, which is a fission yeast, the regulatory protein is CDC2. In remaining all eukaryotes, the maturation promoting factor is named as CDK1. In budding yeast, it is named as CDC28. In fission yeast, it is named as CDC2. In remaining all eukaryotes, this MPF factor is named as CDK1. Discovery of cyclins in sea urchin embryo. Later, some researchers they took the cytoplasm of sea urchin embryo and analyzed the protein. They found some proteins are not in constant in different phases of cell cycle. They are continuously going up and going down in different phases of cell cycle. Therefore, they named these proteins as cyclins. Initially, they discovered only two proteins and named them Cyclin A and Cyclin B in sea urchin embryo. These Cyclin A and Cyclin B are constantly changing in different phases of interphase and different phases of mitosis. Structural elucidation of MPF from frog embryo. Then researchers purified this maturation promoting factor and they found that this MPF comprises of two proteins, cyclins and CDKs. Later it is identified that these cyclin proteins are very important for the CDK activity, and this CDK has kinase activity. Cyclin proteins they phosphorylate CDK protein and make CDK protein active. CDKs can be phosphorylated at three places. One is at threonine 161 amino acid residue. The second one is tyrosine 15 amino acid residue. The other place of phosphorylation is at threonine 14.
Phosphorylation of threonine 161 in this protein makes this protein active, but phosphorylation of tyrosine 15 and threonine 14 makes this protein inactive.
Binding of cyclin B makes phosphorylation of these three amino acid residues. In mitosis itself, these cyclins are degraded by APC complex, that is called anaphase promoting complex or cyclosome. I am going to explain about this APC and cyclosome complex and the regulation of cell cycle by APC and cyclosome complex in mitosis. After proteolytic degradation of cyclin B's by ubiquitination with the help of APC complex, that CDK become inactive. Again this CDK forms a complex with the cyclins at G2 phase in next cell cycle. Cyclins and CDKs in cell cycle regulation. We have seen that cyclins and the CDKs are important for progression of cell from G1 to S phase in some cells and in G2 to M phase in some cells. Then researchers identified that different phases of cell cycle has different types of cyclins and CDKs. I am now going to explain that different cyclins and CDKs in different cell cycle phases. G1 phase has G1 cyclins and G1 bar S cyclins. G1 cyclins activates genes that synthesizes this G1 S cyclins. G1 S cyclins are important for progression of cell from G1 to S phase. Then S phase cyclins. S phase cyclins are important in S phase. S phase cyclins involves in DNA replication in S phase. Then M phase cyclins. Then M phase cyclins. M phase cyclins are synthesized in late G2 phase, and these M phase cyclins are important for progression of cell from G2 to M phase. In M phase there is another protein that is called anaphase promoting complex or cyclosome. This anaphase promoting complex is important for progression of cell from metaphase to anaphase. On the other hand, different CDKs are also present in different cell cycle phases, and these different CDKs forms complex with cyclins of that cell cycle phase. G1S CDK forms complex with G1S cyclins. S phase CDK forms complex with S phase cyclins. M phase CDK forms complex with M phase cyclins. Different cyclins and CDK families in cell cycle regulation. I am going to explain different cyclin and CDK families in cell cycle regulation. I am going to explain in vertebrates and in budding yeast. In vertebrates, 4 cyclins are present. G1 cyclins are called cyclin Ds, and G1S cyclins are called cyclin Es. S phase cyclins are called cyclin A, and M phase CDK are called cyclin B. So these 4 cyclins are present in vertebrates. In G1 phase, cyclin D. In G1S phase, cyclin E, and S phase has cyclin A, M phase has cyclin B. And the CDK partner for these cyclins are 4 CDK partners are present. CDK4 and CDK6 forms complex with cyclin D in G1 phase, and CDK2 forms complex with cyclin A in G1 S phase, and CDK2 and CDK1 forms complex with cyclin A in S phase. CDK1 forms complex with cyclin B in M phase. In budding yeast, only one CDK is present and 4 cyclins are present. Cyclin 3 forms complex with CDK1 in G1 phase. Cyclin 1, 2 forms complex with CDK1 in G1 S phase. Cyclin B5, 6 forms complex with CDK1 in S phase. Cyclin B 1, 2, 3, 4 are present in M phase, which forms complex with the CDK1 in M phase. Regulation of CDKs. CDKs can be regulated by cyclins.
The images of this video lesson are uploaded from below reference books. The Cell, A Molecular Approach by Cooper, Molecular Biology of the Cell by Bruce Albert. Thank you for watching this video, and please subscribe my channel JRS Learn Life Sciences for more updates.